Dr. Joseph Gleeson from the University of California, San Diego and Rady Children's Hospital and the Genomics Institute. I'd like to introduce you to our new paper in PNAS entitled TMEM161B modulates radioglial scaffolding in neocortical development, and introduce you to Dr. Wang and David Sievert from our lab. Polymicrogyria is a disorder of cerebrocortical development in which there are an excessive number of cerebrocortical folds. This associates with epilepsy and neurodevelopmental delay. There are several known causes for polymicrogyria, abbreviated PMG, and they implicate specific cellular functions such as cortical patterning. On the brain scan, one can see the increased number of cortical gyri. From our cohort of nearly 10,000 families in the Middle East, we identified 4 families with a nearly identical form of PMG with bilateral symmetry and severe neurodevelopmental delay. The double bars in the families indicate consanguinity, and that together with the recurrence in families suggested a recessive mutation. This work was in collaboration with Dr. Mahazaki and Dr. Mahmoud Isa from Cairo. All the patients showed homozygous mutations in TMEM161B, and this protein encodes for an 8-pass transmembrane protein in which the patient amino acids were highly conserved across evolution and were located and adjacent to transmembrane domain. We found that TMEM161B is expressed in most of the fetal brain cell types from Nowakowski's 2020 single-cell paper. We found expression in neural progenitors, mature excitatory neurons, inhibitory neurons, radioglial cells, and outer radioglial cells.
To understanding the mechanisms, we generate brain organoid with iPS cells of patient and parent and found unusual bubbles tags in patient's brain organoid but not in healthy control. When we sectioned this organoid and took a detailed look into it, we observed patient's brain organoid. They were disorganized and lack polarity based upon ZOL1 staining, pointed with a white arrow in this picture. In developing human brain, there are a population of radial glial progenitor cells that usually position their cell bodies at the apical surface and extend a radially processed fibers to the basal surface that expressed BLBP, a lipid binding protein. We next stained for the BLBP in brain organoid and found TMEM161B patient-derived brain organoid show disrupted radioglial fibers, suggesting altered polarity.
To understand if the mutations we found in humans were partially or fully inactivating, we collaborated with Dr. Stephen Murray and Caleb Hefner from the Jackson Laboratories, who generated patient mutation crispin knock-in mice. They successfully generated an allelic series including genotypes minus, plus, as well as knock-in alleles. They were harvested at embryonic day 18.5 to assess the phenotype. We found that all crispin F0 genotypes were grossly normal, except for the loss of function over the loss of function, which showed severe defects in the head and rest of the cranium that showed an otocephaly-like phenotype. This was reported previously in mice and consists of a retrognathia as well as skull defects in microcephaly. Gross brain analysis showed normal appearance of the allelic series, except again for the loss of function over the loss of function, where half of them showed defects in the hemispheric midline cleavage consistent with holoprosencephaly, indicated by these arrows on the far right. While the brains of the loss of function over the loss of function showed severe defects in cerebrocortical development, we analyzed more carefully the knock-in over knock-in, these are containing the patient mutations, and we found mispositioning of the SAPB2 superficial layer cells, suggesting a defect in radiomigration.
What is the function of TMEM161B in cell? To understand the function, we first expressed TMEM161B in glial cells and was surprised to find dramatic actin spikes that co-labeled with fluoride. We further confirmed that plasmid containing patient mutations were defective in this phenotype. Furthermore, we found TMEM161B biograde analysis predicted interaction with a cohort of cytoskeleton proteins, including Rho-GTPase CDC42. CDC42 was previously implicated in actin polymerization and holoprosencephaly. You will find more details in our paper regarding how do TMEM161B modulates actin filopodia spikes through CDC42.
Together, the models suggest that TMEM161B is required for apical polarity, functioning upstream of CDC42 in actin polymerization. To conclude, loss of TMEM161B leads to polymicrogyria with severe seizures and neurodevelopmental delay. TMEM161B is required for apical polarity and migration in the developing brain, and it functions upstream of CDC42 mediating actin polymerization. We'd like to acknowledge our numerous collaborators and funding sources. Thank you for the opportunity to present our work.